Selaginella, and Isoedes. The club mosses are Lycopodium, spike mosses, Selaginella, quillworts, Isoedes. So here we have, what's this one? Remember this guy? Do you still have it, Sarah? Okay. Oh, you can use it. All right. What do you guys think? What is this one? So you are going to need, for the exam, you are going to need to be able to recognize these different plants we're learning about. Any guesses? If you guess wrong, don't be embarrassed by it. It's okay. What do you guys think? That's an awesome guess. But this one is a club. This is the club moss. And club moss and spike moss do look a lot alike. So that's the tricky part. So what I would do, to be fair, is on the exam, I would probably also show you a close-up of what's happening inside of the strobili. Because if you remember, one of them's heterosporous, one of them's mosporous. So if you see a close-up of the strobili, you would be able to tell, ooh, that one's heterosporous, and the heterosporous one is, remember that? Is Selaginella, which is spike moss. The one that's mosporous is Lycopodium. That's the club moss. They do look a little different as well. That's Lycopodium with the flammable spores. And here is a longitudinal section of its strobilus. And notice other than this little weird artifact glob thing right there, all of these have microspores in them. Okay, so homosporous. Then we've got Selaginella, which does have a similar look to it. And here is the strobili. They don't shoot up as high as we see with Lycopodium, and Lycopodium does tend to stand a little taller. Selaginella is a little more prostrate growing. All right, but then if you see the strobilus of Selaginella, you should be like, boom, I know what that is. Heterosporous. There's two different kind of spores being developed there. Okay? You guys with me on this? All right. Okay. And then we've got, come back here, the other example is within Lycophyta, quillworts. Isoedes. Do you guys remember what that one looks like? Quillwort. It's like a little porcupine, the quills, kind of, right? But a wart version. Remember, wart is like the Old English term for like an herb, a medicinal, generally medicinal kind of herb. And here is our example of one. And they are heterosporous, and they produce their sporangia down at the base of these microphylls. Okay. Then there's just two other major groups that we learned. We have Equisetum and the true Pteridophyta, Polypodiophyta, and the true ferns. Okay? So scrolling back down to this part of the lab, we had some examples, living examples that didn't look this great, but here I'm showing you examples of fertile stalks with the strobili, those interesting little whorled leaves, ribbed stems, really rough to the touch, snake grass, horsetail are common names, and check this out, guys. Remember the strobili of Equisetum have these really cool sporangiophores, these structures that extend out and then develop multiple sporangia hanging off of them. And those spores do have, do I have a close-up of them? No, I don't. If you were to look really close here, that's too blurry. You would see that they have those cool little structures that help those spores shoot out really fast. Do you remember what those are? Elaters, good job. So the spore is like, it's almost like it's got little arms. It's hugging itself. When it's moist, when it dries out, it's like, I'm gonna hug you. And it opens its arms up and they shoot out. And then ferns, a lot of diversity in ferns, and you got to see some different living examples last time. This is showing different kinds of, what's it called when their spores are grouped together on the backs of the fern fronds? What are those little groupings called?
Sori.
Sori. A single one would be a sorus. And when you look really hard, sometimes your eyes get sore. They give you sore eyes when you're trying to look really close and see the individual sporangia, right? And this one has indusia that fold over and protect the sori here. All right, and let's go down a little further, zooming in on a single sorus that does not have an indusia. Those are like the ones we saw in class. A lot of them look like this. And we zoom in on a single sporangium, and we see that they have this really cool little mohawk structure, the annulus. And as that dries out, it breaks open these little cells here, the lip cells, and the spores shoot out. Today, we do have Azolla today, the cute little aquatic fern. So you remember we talked about this last time, that when you take these little tiny fern fronds, they're so cute. They're like tiny, and you're gonna take a little bit of that, put it on your slide, a couple drops of water, cover slip on top, and then you do this thing where you like squish, and you squish it. And what you're doing is you're just squishing open those, the cells of the leaves, and in doing that, there's little areas within the leaf cells where the cyanobacteria are living. And you'll squish out cyanobacteria. So you'll see cyanobacteria, really cool little green, they look like green pearl necklace or beaded necklace, but green. And they're pretty cool. We'll see those today. Then fern babies. The fern gametophytes look like little hearts, unless they send out hormone triggers to cause, or kind of like to stop the growth of other gametophytes so that they only develop antheridia. The heart-shaped ones or mitten-shaped ones, the ones that have the two main lobes, are bisexual or hermaphroditic. They have both archegonia and antheridia. So remember, archegonia have those neck cells, and they're kind of hanging down toward the ground. And the antheridia are little bulbous structures with sperm inside. The sperm come out, swim up into the archegonia to connect with the egg inside the venter, right in there. And then you're going to go through that process and add the terminology in. The end, good story. And then that spore, the zygote then develops into then that next sporophyte generation to then produce spores. All right, that's a lot of stuff we covered. And so now we're taking a break and stepping back a little bit, taking a break from plants and jumping into other interesting organisms that are plant-like. So they also photosynthesize. They may or may not be green. They're often green. And some of them have really intimate relationships with plants, cyanobacteria, either in the form of just the fact that the chloroplasts in plant cells evolved from cyanobacteria. That's a unique connection, right? And there's cyanobacteria alive today that look really similar to chloroplasts in plant cells. And then also we have plants that harbor, that produce houses for all kinds of bacteria. Cyanobacteria or non-photosynthesizing bacteria. So they have that close link. And then green algae, they are similar. to plants, and so it's important to talk about those other photosynthesizing organisms. And a lot of people will say like plants of the sea, or throw green algae and red algae and brown algae in with the plant group, but they are not in kingdom Plantae. But they're still important. And if we don't talk about them too much here, you might touch on them if you take a higher level biology course, or even a lower level biology course. And you might talk about them in zoology as well, but we take a little extra time to talk about them here. So first off, connection between bacteria and plants. So here I have a picture of the Azolla fern that you're going to get a chance to look at up close. And inside those leaves, you'll see cyanobacteria looks like this, a little green beaded necklace. What is this picture showing? Do you remember? Well, first off, what does it look like? What is it? They're roots. Okay, good. Roots, and what's happening on those roots? Do you see anything interesting? Nodules. There's these little bulbous structures here. So these nodules are little houses for bacteria to live. So this is what the roots would look like. We would see these on the redbud that we keyed out the other day, and the American vetch. So not just the legume family do we see these, but pretty much everything in that family has this kind of relationship, which is pretty cool. And then, like I mentioned, chloroplasts evolve from cyanobacterial cells. So lots of interesting connections between bacteria and plants. And these are all positive examples. There's also negative bacterial plant associations, and plants can get bacterial infections like we can. And just like we also harbor a lot of really good bacteria in and on our bodies, plants do as well. So going back to diversity of life on Earth, and we already kind of went over this, but just a reminder, we've got domain, all of life on Earth can be split into 3 domains, and then if we select one of those domains, we'll see that we can split the organisms in that domain up into different kingdoms, and then different phyla, and so forth, so forth. And if we look at here at all, this is a family tree showing all of life on Earth. The root of the tree right here would be where our hypothesized common ancestor of all of life on Earth would be. Showing that the ends of all these branches is showing living organisms, and that they all are united, connected together, and share a common ancestor. And we have these major branch out here, which is domain bacteria, which is very diverse. And then we have domain archaea, and then domain eukarya, of which we find plants, fungi, Kingdom Animalia, and then protists. So, so focusing in, bacteria are considered prokaryote. They don't have a nuclear envelope. They don't have, they're not as complex as eukaryotic cells, right? They don't have all those organelles that we've learned about with plant cells. And they don't just make up bacteria, it's also archaea, which are 2 very different groups. But they are, we know of already, they are tiny, but there's a lot of them on Earth. And if we think about the ocean and just thinking about, you know, the water, it might look a little cloudy, we're not sure what's in there. There's a lot of microscopic organisms in there, and a lot of that is made up of bacterial cells. Same thing, if you have healthy soil will have a lot of microbes in it as well. And our mouths have a lot of stuff, a lot of bacteria on them, some of which are good and some not so good, but it's all over the place. So these are thought to be the first fossils of life. Are most closely resemble cyanobacteria, and specifically a species that you're going to see today. It looks like this species, and it's called Oscillatoria, and it is made up of these little tiny segments forming a colony, and so these are, and this one's been dated to be 3.5 billion years old. Cyanobacteria. So going through, I'm going to be focusing on bacteria, but just to let you guys know a little bit of the difference between archaea and bacteria is that they do have very different things happening with cell walls between the two groups. Different kinds of lipids are produced between archaea and bacteria. Very different DNA and RNA sequences, so they've been separated from each other for a very long time evolutionarily. A lot of differences there, and they have different metabolics. Archaea, if you hear the term extremophiles, there are some archaea that can live in very salty, very acidic, and very hot environments. So these would be the critters growing in like hot hydrothermal regions in Lassen or Yellowstone, and causing those really cool colors in the veins. Yes?
So how would they, like those extremophiles, get to all those places? Because I feel like those places are few and far between. So how would they, like...
That's a really good question, like thinking about, yeah, how does anything get around, right? Well, think about how do you think those guys would get around?
Water? I don't know.
Yeah, they can get around by water. Also, there are certain life stages where there'll be like a kind of a resting spore kind of stage, or a spore where if the area dries out, like wind can pick it up and they're just become part of dust.
Oh.
And you can imagine that can get up high in the atmosphere and can go travel around to the other side of the world. And so it's kind of crazy thinking about that, but there's stuff all over in the air right now. And if it lands in a suitable location, it can start to multiply. Did I see another hand? Back? Question? Comment?
No,
Yeah.
I was just gonna see, like, wouldn't some like go in the area like evolve to survive those conditions, or would they have evolved before they went into the area?
That's a really good question. I think it depends, and both things are going on. You get some things that might have a certain characteristic, evolved some sort of trait that is unique for that population, and it happens to end up in another place that new characteristic that's kind of unique for its group didn't really do anything for it here, but then in a new location it might actually help it survive. And or you can have something that's able to survive, it's there, and then a new mutation occurs that can actually help them do even better and start to take over more of that space. So yeah, both kind of situations could potentially happen. And if they don't have that trait, the traits that help them live in a new area, they just wouldn't be able to pass on their genes. And it was like, good try. Didn't work for you though. And some can even live off things like inorganic substances like sulfur. All right, that's it for archaea. Just know that there's this other unique, interesting group that encompasses part of that prokaryotic designation. Yes?
So what's the actual difference between bacteria and archaea?
There's multiple differences. So I mentioned we've got a lot of different kinds of things. Because at one time, before scientists started really looking at this stuff, especially before scientists were comparing DNA, they were grouped together. They're just like, these are unicellular organisms that don't have nuclear envelope. They're prokaryotes. They're just like, they're their own group. And they were called bacteria. And then you have then that split into archaea and eubacteria. EU for true bacteria. And now I think it's just called bacteria and then archaea. But there's been it's been, at one time they were grouped all together. And then there was also another term maybe you've heard of called Monera, of which bacteria and some other things were thrown in. Yeah?
Where do viruses fit into it? Because I know there's been a lot of debate over whether or not they're actually should be considered alive.
Yeah. Well, do you think they can fit into anything that we're talking about so far? It's tricky. Well, the viruses don't have their own cells. So, everything that we're talking about is either a single-celled organism or they get together and form a colony, or they're multicellular, which is like a very advanced colony. We're like an advanced colony of lots of cells. So we're multicellular, but we have a lot of other things living on us and in us too. So it's a really good question. Viruses, they don't have the smallest unit of the accepted term for the smallest unit of life is a cell. And so if they don't fit into that category of life, if you use that definition. But yeah, there's a lot of folks who feel like, well, they sure act like they're alive. And they're doing it without a cell. They're making use of other cells. And so they're in their own category, viruses. But they have DNA, they have RNA, and they're proteins. That's what a virus is made of. It's proteins, DNA, RNA, that kind of thing. Yeah.
Never mind, I answered my own question.
Oh, okay. Good job. All right, so on to bacteria. I mentioned already they're everywhere, outnumber eukaryotes.
Right.
They can both cause disease or be beneficial, depending on what they are, where they are, who they're in, and what's going on. Decomposers, so really important in the world. And this is just interesting imaging showing bacterial evolution through antibiotic resistance. So you can actually see evolution happening on a petri dish or plate, like you see here. So this is showing bacteria that run into antibiotics, and then you see changes. So these different colors are showing that the DNA sequencing is changing. They're actually diversifying. And some aren't able to live with the antibiotics, and then some can, and then more can, and more can, and more can. So you can see why it's good to not overdo antibiotics, right? Because you're helping to support that antibiotic resistance if we use too much. Yes?
So they put some bacteria in a petri dish with some,
With antibiotics.
and it was the same species at the start?
Yeah, and then they allow them to grow and reproduce and keep moving on, and it kind of growth slows down, and then some of them survive, and then growth speeds up again, and so forth.
So are they different species by
It's
the
like,
end?
and I have to go back and double check with this, specifically how they had this experiment set up, whether or not it was different concentrations of antibiotics as the bacteria progress through the process, or if they're using different antibiotics. I have to double check on that part, but it's showing that that you can see that the bacteria are evolving and changing as the growth of the bacteria spreads. And so this is then just continuing with that, just showing the original organism is the light gray with low resistance, low antibiotic resistance. And then you have these ones, so these three different branches are showing from the same species of bacteria, you're seeing that of them, there's different kinds of mutations that are occurring that are causing them to be able to be resistant. And it's showing intermediate resistance. But there are different kinds of mutations that have occurred to allow for this, so that's why they're showing the 3 different branches.
Oh.
Along this branch, you see that there's even higher resistance. So, so they're continuing to grow and develop, whereas here we see this one it petered out and it lost that resistance actually. Question, comment?
What is a block?
What is what?
it? A blocked mutant?
A blocked mutant?
Yeah.
Looks like that's the one.
Like
that's
light
by the
brown.
blue.
Yeah, looks like that's this one right here. Yeah. So,
What does it mean?
It was just blocked from going any further, but.
that one, to me, it's been a little while since I looked at this, is that it lost that ability to be able to be resistant to the bacteria anymore. So you see that you've got, mutations will occur that are allowing some of those branches of these, as they're dividing and spreading apart, some of them might have that mutation, and then those then are going to reproduce and reproduce and reproduce, right? So they're passing on that mutation, but you might have then some that don't get that mutation passed on to them, and then they're going to reproduce. And so then all of a sudden you have some, like right there, where that mutation was blocked. It's not showing up anymore. Okay. And so they're not able to have that resistance. So first off, this is a reminder of what we learned before. Big relationship between plants and bacteria is first of all, the engulfing endosymbiosis, right? If we're talking just bacteria that don't photosynthesize, so bacteria that take in oxygen and eat other things, you have a cell that did that. It engulfed something else. It engulfed another unicellular organism similar to itself, and instead of eating it, it allowed it to live inside, and it got food from the cell it was living in and was able to make extra ATP that it then shared with the rest of the cells. And that was what?
Mitochondria.
Mitochondria.
Yeah.
So mitochondria would be that bacteria that was engulfed and taken in and allowed to live inside there. And then we've got one branch of that. So here we've got bacteria, aerobic heterotrophic prokaryote being engulfed. We can have that line continue on this way, and that's our line, kingdom Animalia, and other organisms that don't photosynthesize. And then we have here another cell was engulfed, this one cyanobacterial cell, and that then became chloroplast. So that's the first big link between plants and bacteria, both the evolutionary history of mitochondria and chloroplasts, which is all review for you guys because we covered that already. And so I'm just going to go through a few examples of relationships that plants have with bacteria, and mostly emphasize the good ones. There's bad ones out there too, causing bacterial diseases. But this is the one we've already been talking a bit about, is nitrogen-fixing bacteria living in special root nodules of certain species. Not all species do this. Species that don't do this can take advantage of species who do if they live nearby. If their roots are able to get close to, if another plant's roots are able to get over here, some of that nitrogen that's fixed can get absorbed into their roots. So nitrogen-fixing bacteria. And something I did notice on the exam, there was that question, you remember, what can plants do when they live in harsh environments to be able to get nutrients that they can't get, like nitrogen? And most of you guys did awesome, just like remembering carnivorous plants, that's a big one right there. And then some of you said this, nitrogen-fixing bacteria in the roots. And then some folks talked about mycorrhizae. So remember, mycorrhizae, they do help plants take in nutrients. So more like minerals along with the water. But fungi are not nitrogen fixing. So fungi aren't going to provide that nitrogen fixation. So remember that. That's a big difference if you're thinking about the difference between these cool relationships between fungi and plants. They're helping absorb water and minerals, the fungi are, whereas the bacteria are helping with nitrogen fixation, okay? This is how that process works. These bacteria that live in Fabaceae plants, so bean, legume plants, like redbud and the sweet vetch, we talked about lentils. That is a genus Rhizobium that does that. So you've got the nitrogen in a gaseous form. Plants cannot take that in. We can't take that in. We're breathing it right now. It's just we're breathing it in, breathing it out. Nothing's happening. We have to get our nitrogen by eating it. And the stuff we're eating gets it because of plants. Plants were able to get it inside them by bacteria. Some bacteria just might live in the soil and do it, and plants can take advantage of it, or they are living in their roots. And so the bacteria have a special enzyme that plants don't have, that we don't have, the special nitrogenase enzyme, to change that structure. And it's called fixing. It's converting a gas into a solid. And so it fixes nitrogen into a solid form. The plant can take that in. The plant can use that and pull it into its proteins, its amino acids. It can pull it into when they're making their new DNA and RNA molecules, and use that nitrogen in all those ways. Nitrogen's a part of chlorophyll pigment molecules and a lot of other important molecules. And then when we eat it, right, those high protein plants like beans, they have a lot of extra protein in them because they're able to just take in so much extra nitrogen. And then looking at the nitrogen cycle in general, we have nitrogen fixing plant, that's bringing nitrogen into the soil, and then there's some bacteria just living free in the soil that are fixing nitrogen. And we've got plants can pull that in. And then you've got plants breaking down, animals breaking down, and the detrivores can break those down, and that pulls also more, can bring more nitrogen into the soil as well. Like fertilizers, natural fertilizers. Or if we're adding artificial fertilizers, it adds it too. This is another plant that does something really cool like this, and they're called alder plants. Are you familiar with alders? Mm-hmm. We have around here, we have white alder that grows near creeks. We might see a couple on our field trip. We also have what's called mountain alder, and then on the coast, red alders are all over the place out there. And you can recognize them by their leaves. They have these really cool doubly serrated leaves that look like this. You have, first I'm going to do the big serrations. And then each of these then, if you look close, you can see that these are actually made up of multiple serrations. Each little serration is made up of even more serrations. So that would be called doubly serrate, or doubly serrate, or doubly toothed, you might hear it. Those are alders. And in their roots, they form nodules. And it's a different genus. This one's called Frankia. So a different genus of bacteria. All right, wait, what are the bacteria getting out of this? I almost forgot about that part. Yeah, that's important, right? The bacteria are getting something out of this. They're providing the nitrogen for the plant, and these are bacteria that need to eat. They're heterotrophic, and the plant is giving them food. So, good relationship. Here are some bad bacteria examples. We have Agrobacterium, which, if you remember, Agrobacterium is that bacteria that is used in genetic modification. That's the one because it can already in nature get into a plant, put some of its own DNA into the plant's genome. It does it naturally, so we've made use of that. Then so here's some other examples of different bacterial infections. And then right down here is a good one. There's that one good example, the rhizobium. So here's Agrobacterium, crown gall disease, that we use in genetic modification. It produces these tumorous growths on the plants and can kill the plant. So when I'm talking about prokaryotes being good for the environment, I'm talking about a lot of different ways that can be good for the environment. We've talked about some examples, but a really big one is breaking down dead things, so that nutrients can be recycled and used in different ways on Earth. And we have made a lot of them work for us in different things like water treatment centers, waste treatment plants, and my computer just died. And actually, so I've got a natural history field trip. We're leaving tomorrow for the weekend. And I'm hoping we're gonna have some time to visit the Arcata Marsh. Has anybody been there before? You have? Okay, cool. So you see that they have a really cool setup there with their water treatment center. And it's all natural, using microbes and then using plants to filter all the wastewater from the area. Are you gonna wake up? Okay, it's not gonna be long to wake back up. But it is asking for my password. All right, so that's called bioremediation, using living organisms to remove pollutants from the environment. So some folks would use them for biological control. GMOs, right? Using bacteria that, not only using bacteria to get genes into new genomes, but also using bacteria that produce proteins that are toxic to insects, using that section of DNA to then make it so the plant is producing that protein, like we see here with the BT plants. And a lot of all living organisms, multicellular organisms, they have relationships with prokaryotes that help them out. And you guys all know how they help us, right? Probiotics. If we have too many antibiotics in our systems, we can kill that good flora. And so we need to bring it back in by eating good things that have good probiotics in them. Also using bacterial waste products, ascorbic acid, vitamin C, that's a waste product that we use from bacteria. And added to a lot of foods. So obviously having the good stuff, if you start getting low on the good bacteria, then the bad stuff can come in and start building their homes in areas where you don't want them to. And that waste can build up. So cyanobacteria, and we have a few live specimens we're gonna look at today. One common name for cyanobacteria is blue-green algae. But that's one of those common names that is confusing, right? It's not algae. They should have called it blue-green bacteria, because they are bluish-green. But if you just say cyanobacteria, that is actually a good way to go, and a lot of people nowadays know that term, I feel like. But most people are familiar with the blue-green algae, because people use it like a dietary supplement. Anybody ever used it before? Blue-green algae, more bacteria. Spirulina is a common one people have added. So here we have cyanobacteria. If you look close zoom in on cyanobacteria, you'll see the thylakoids. very similar to what you see, what you learned about in chloroplasts. They have thylakoids. They obviously will have their circular DNA, and they're pretty much free living chloroplasts in different kinds of shapes, is what cyanobacteria are. So they have, these are special roles that, the characteristics, talents, I should say, that cyanobacteria have. One big one is they produce oxygen. Another one, they produce food for themselves. That's all through photosynthesis. They also can fix nitrogen. So they have pretty special roles in the environment along that. They do this with the help of, for photosynthesis, chlorophyll A, but they don't have chlorophyll B or C. They have chlorophyll A. And then they also have other important pigments. So other kinds of bacteria that we've talked about, like that make a relationship with bean plants, alder plants, they do not have, they're not cyanobacteria. They're not able to do this. These are some early fossils. And they're rocks that are actually just made of mats of cyanobacteria. So cyanobacteria and bacteria in general, they form a matrix. If you guys have ever had that experience where you're camping for a couple days or something happens and you don't get a chance to brush your teeth as often as you'd like, and you start to get that film on your teeth. You guys know what I'm talking about? And people will say like, fuzzy teeth or whatever. That is actually the bacteria on your teeth are producing this matrix. And it's like this kind of slimy stuff. So some bacteria will form like this big gelatinous ball. And we will actually, one of the green algae or cyanobacteria that you're going to look at does do that. It's called nostoc. And these are fossils of cyanobacteria that form these mats, these flattened mats of just themselves with all this interesting slimy matrix around it. And then it'll be layers and layers and layers and layers of growth. And then it becomes actually a sedimentary rock. So that's what and that's called stromatolites. And some parts of the world, you can see new ones where they're getting formed, and then they've dug them up, and that's where some of the oldest fossils that we found are from, things like that. So not only do they look like some cyanobacteria that we see living today, but they were found in that layering kind of growth that certain cyanobacteria can produce. So do you guys see how there's green and then there's this yellowy color? That yellowy color is showing that matrix I was talking about, that slimy matrix that some bacteria will produce. So specifically, this is nostoc. That's the one I just mentioned we have a sample of. They form these like gelatinous little balls or blobs, and inside are these green little chains. These are the individual cells, and they form chain colonies. And we see a lot of different types here. Okay, looking at this, do you see one that reminds you most of this fossil? Look at the fossil, and then look at this image. Which one? Can you guys read it from where you're sitting? I don't know if you can.
Middle left, Annabelle.
This one?
Yeah.
It does kind of look like that. What about this one?
Nah.
One of those two? Yeah, it kind of looks, it does. It has that look to it. So gelatinous sheets or, biofilm. That's what that material is actually. That's one of the names for it. Maybe you've heard that before, biofilm. All right, so spirulina is a classic one, and it has it's a spiral shape to it. Oscillatoria has a sheath, a biofilm kind of sheath, and then they call them trichomes here, but really what it is just like a row, like rows of cells within this gelatinous sheath. That's oscillatoria. down here. How many L's? 2 L's. Oscillatoria. And it's thin. These filaments are like as thin as a piece of hair. They're thin. So when you look at this one under the microscope, you're gonna wanna try your best to go to the highest power you can and really zoom and really mess with your fine tune. So then you're gonna see, and you're gonna barely, when you've missed the fine tune, you're gonna barely see these little tiny divisions within. And those are the cells. That's oscillatoria. And you get to the end, it does have a little bit of a rounded look to those ends. Spirulina, oscillatoria, so oscillatoria is one of the ones that they think is not actually that fossil. You can't say that that fossil is oscillatoria, but it's something that looks very similar to it. Or Anabaena as well as another one. So Nostoc and Anabaena, you're gonna see both of those today. Nostoc and let's see, let's put Anabaena. So Nostoc is will kind of look like a little gooey ball kind of structure, and that's that gelatinous biofilm. And then other than that, and then all through it, it's got long chains of little beaded, they look like little green beaded necklaces. Whereas Anabaena, when you find Anabaena growing, it's not in those gelatinous globs. It's on its own. So these would be considered vegetative cells. And with Nostoc and Anabaena, you'll also see two other kinds of cells in those colonies. And I'm gonna do them missing. You're gonna have a larger one that's kind of like very translucent looking. And then you're gonna have one that has a little bit of a different shape and is maybe darker. All right, so can you guys tell? Those are different looking cells, right? One of them, they have different functions in this whole relationship. The larger, clearish looking ones are called heterocysts. And then the other one, which is a little darker, a little bit elongated in shape, is called an aconite. So, and they have a little bit different functions. The aconites are thicker walled cells that if it becomes really dry and then it dies, everything will die except for those aconites become like a spore kind of structure. So that when environment's suitable again, it'll start dividing and dividing and dividing and form a new colony. The other one, the heterocyst, these are nitrogen fixing cells. So nitrogen fixing is hard work, and that's their job. Those cells, they don't photosynthesize, they focus in on fixing nitrogen. So what do you think all these vegetative ones do? All the other ones that are smaller and green? They photosynthesize. They're making food for everybody. So, so this would be considered a filamentous colony, is how those are. They are unicellular, but they do come together and form colonies for a large part of their life. All right, so here are some examples of nostoc. So remember, nostoc's the gooey blob one. So gooey blob, and we do have some samples. But what I recommend is when you guys do this sample, you're going to want to not do a slide of the entire big ball. You're gonna take like part of it, try to take a little bit of it and put it on your microscope slide. So you can share with the rest of the class. Because usually we only get a few of them. But there are some parts of the world where they will, they'll get like this big in some freshwater lakes, and people have been known to eat them. So here you can identify the heterocyst. The heterocyst is still slightly green, but just not as bright green. It'll be more translucent and larger. Now there's a plant that actually develops nostoc in it, and the nostoc helps obviously with nitrogen fixation, and instead of producing these gooey blobs that it produces, it's living inside the plant. So obviously, cyanobacteria is photosynthesizing. It produces its own food, but it can get extra food from the plant, and it has a place to live, a safe place to live, where it doesn't have to worry about drying out. So Anabaena is that next one that looks very similar to nostoc, but it does not form those big blobs. So you can see a difference, aconite versus the heterocyst, and this is the one that you're gonna see when you squish the Azolla fern. The Azolla fern has the Anabaena living in it. So what's pretty cool about these is a common name is mosquito fern, is in rice paddies. This is commonly found. If it's not there, people will put it in the rice paddies. Rice is not a nitrogen fixer. Rice, similar to corn, grasses, they love lots of nitrogen, and they can't make it on their own. So you either have to add a lot of fertilizer in, but if you just add some of these plants in there with the rice, then you don't have to add the nitrogen fertilizer. So we haven't talked about cycads yet, but they're a type of gymnosperm that looks like a palm or a fern. If you know a sago palm, maybe some of you guys might have one growing in your yard. In their roots, they have cyanobacteria living inside. And just an interesting little side story. There's a certain region where scientists were studying why people in this certain region in Guam, why they were getting so many different kinds of neurological disorders, and such as Parkinson's, ALS, dementia, Alzheimer, and this is a really crazy story where you're like, the scientific investigation at its best right here, where scientists were trying to figure out what was going on. They were looking at the diet. They were trying to figure this out. In this region, it was culturally part of their culture to eat these fruit bats, and the fruit bats. So what are the fruit bats eating? Anyways, well, first of all, the scientists were looked at the fruit bats and looked at whether or not they were harboring toxins, and they were. They were harboring a lot of this, what's called BMAA, beta methylamino L-alanine, which is a neurotoxin. And a lot of different organisms will produce neurotoxins. You've probably heard of, like, you don't want to collect shellfish a certain times of the year on the coast, or you'll have a sign like, don't eat the shellfish. There's certain organisms in the ocean that are producing neurotoxins, and it can harbor in certain animals. Well, they found that it was happening with these bats. So then they were looking at, well, how are the bats getting into it? And it didn't seem like it was affecting the bats too much. And then they looked at what the bats were eating. The bats were eating the seeds of the cycad. And the people actually ate the cycad seeds too. But when the people eat the cycad seeds, they'll dry it and they'll crush it up, and then they will rinse it multiple times with water to get any toxins out. And but with the bats, they're just eating the bats. And what happens, there's something that's called bioaccumulation, where an animal, a creature that has, like a bat with muscles, they'll eat toxins, and the toxins could potentially build up in their muscle tissue and maybe not affect them. But then if something eats it, then they're getting really higher amounts than if they just ate the seeds from the plant themselves. And so they found that they were eating these seeds, the bats were eating the seeds, and they found the toxins in the seeds, and then linked it to the cyanobacteria living in the roots. So you've got cyanobacteria in the roots, getting toxins into the cycad, bat eating the seeds of the cycad, and then people eating the bats. And then people getting a lot of these really high numbers compared to other parts of the world of these neurological disorders. So in the end, guess what? The bats are actually in danger of being overhunted, and also habitat destruction is a huge one, so that was probably linking to it as well. But then eating them wasn't very good for their population. Another common name is flying foxes, because they're all furry and they fly, and they're mammal, right? So they're fruit bats. Yeah, there's some that can get there's some that can get really big too. They're really cool bats, and they're fruit bats. They love to eat fruit. So this was kind of an easy way, because it's kind of like we come in, you have people come into a culture and be like, hey, you shouldn't do that, and it's their culture, and it's like, who are we to say you need to change your culture, right? And we did that a lot in not we, but our ancestors. A lot of our ancestors did that. In different parts of the world, you come in and you kind of take over and say, you have to do this way. So, but this was being able to say, hey, what you're doing, we know it's culturally important and it's part of your culture, but it's making you sick. And this is why. And being able to explain that, and then also that the bats were having a hard time anyways, and so it helped protect the bats. So it's an interesting story, all related to multiple different groups of life. That's not the total end of this story though, because the scientists who were involved with figuring out that story, or that mystery, they went on to study different areas of the world where there's cyanobacteria. Like, let's say you live by a lake and the wind hits the side of the lake where you live. They were starting to find links to people being in these areas having higher rates of neurological disorders, and linking it to cyanobacteria back to, or if you swim in areas a lot, or different things like that, and started thinking like, is cyanobacteria good for us to be eating? Are people who are putting the cyanobacteria in supplements, are they testing for BMA? And so these are big, look it up. It's really interesting. If you like to have cyanobacteria as a supplement, double check that, you're getting some that is good and not full of toxins. Cyanobacteria in sloths, they have been found to grow in sloths very commonly, and kind of help with them looking more like lichen, like the lichen that they're climbing, the trees that they're climbing on. And if you've heard of crusty soil, cyanobacteria makes a big part of what's called cryptobionic soil, and in some desert regions, that stuff takes a long time to be made, and it helps to provide a place for living things to live in the soil, and protects the soil, and people will if you step on it, you ruin years of growth. And so if you've ever been to some of these areas, they'll talk about it, tell you please walk on rocks or on trails, and they'll just start tramping through. And when it rains, then they all kind of come alive and do their thing for a while, and then they dry up. So, cyanobacteria, there's a lot of stuff here. This is just recapping what we talked about. This is a big one that I hadn't mentioned too much, but those first organisms on Earth that happened to be photosynthesizing organisms really built up that oxygen and helped make it possible for more things to evolve and life to be possible the way it is now. Yes?
Didn't that buildup of oxygen cause like an extinction event or something?
So yeah, so critters that don't like oxygen, it wasn't good for them, but it was good for organisms that like oxygen,
That's good.
like us. And then there was a time when there was even more oxygen in the environment, which it would have been probably too much for us, or we would have been a lot bigger. We kind of talked about that last time, didn't we? But yeah, specifically for insects, there's a time when insects and also are huge, and that's during that time period when there was a higher amount of oxygen. So not only is oxygen important for organisms who need it, the ozone layer helped also, that changed things in a way in the atmosphere for organisms to be able to live on land and get out of water. All right, so any questions? You guys want a little snack? Yeah? We'll have a little snack, and we'll go just a little bit longer, and start talking about algae. Then we'll take a break, and then we'll finish that, and then jump into our lab. So we're going to eat some algae while we talk about some algae. If you don't like algae, you obviously don't have to eat it. Okay?
If you don't like algae, get out of this class.
Here, can you help me?
Algae deficient people.
Algae deficiency.
That was snoring.
Let's just keep these cycling around, and people can take one first, and then if it comes through you again, you can take another.
Each package passes by you, so you take one each time.
Dude opened it. You could do that in a sec.
Are those the ones from pasta?
You like that word hard? I didn't know seaweed was considered algae, I'm gonna be honest.
What do you think it was?
I thought it would have been like a, you know how like whales were mammals that like wanted to be fish?
You don't like that nice oil?
Okay.
Plant that wanted to be
How many mammals fly?
Or like it's roasted.
So how many
Here, let's
mammals
get
fly?
another. Let's
There's
get another one
bats.
open.
We got one already.
Yeah, there's one over
Well, I
here.
want to just keep going if people want some.
I want some. There you go.
Here's another one.
Oh, we can grab another from that?
Did you want it?
Wait, where did it come from?
I don't know, he just handed it to me.
Hey, it came around again. I need those.
I remember like a couple years ago, everyone ate seaweed.
The third one. No, third one. Pass it across.
What about like on an egg?
We'll pass it across and say Josh told you to.
No.
He did.
Hey, Sarah's keeping one for
Would you put it
himself.
on hot sauce would be fair.
It's fine somewhere.
Okay, that's some of those have salt in it.
What? Okay.
Do you want your drink when the break happens?
Is it true that white people only eat like unseasoned boiled chicken?
What?
You can get it. Oh, British, yeah, not white people, my bad. The Italians are pretty good at some stuff, I guess. Like skilling with
I saw a sign somewhere, like a question. It was, is British food really that bad? and the top
I think
reply
people
was
should
if
stick
made
to
properly,
pickle
yes.
vegetables and not
I've
eggs.
never tried one.
I'll get some and I'll bring
You know,
Okay.
the beautiful one and delicious cuisine made the Britishmen the best sailors in the world.
Yeah. They ruin it.
They crossed the lanes to find spices and used none of them.
Let's keep going and get the.
Or India.
Eating the algae. Keep that algae going around.
Yeah.
If I was to ask you what kind of algae do you think that is, do you think it's, because there's three major types once we get into the main algal grouping.
The last
There's
one.
red, brown, and green. What do you think is red, brown, and green?
Red.
Green, red, and yellow, right?
It looks green, but it's actually a red algae. I know, it's crazy. Because all the different kinds of algae still have chlorophyll in them, which is green, and other pigments that can sometimes like overwhelm them. All right, back to this family tree of life. So we talked about these, that we went really quickly through those. And now we're jumping into eukaryotes, which we're in Kingdom Plantae right now. We will still be touching on fungi at some point. And then we're gonna kind of talk about this stuff right here. Because they're pretty much, if you look here, right around, somewhere around here is where that branch is off of the other prokaryotes. So we have that early prokaryotic cell that started having a nuclear envelope and was engulfing things. And its membrane was like coming in on itself. We have endoplasmic reticulum starting to form, Golgi apparatus, all this stuff. And then we have engulfing events happening. And mitochondria becoming a part of these cells, and then sometimes we have some branches going off where chloroplasts then develop. But those early ones would have been considered in the protists. And protists are like, don't know where, they don't belong in, they're eukaryotes. They have a nucleus, they have organelles, they have mitochondria, but they don't really match up to be a plant or an animal or a fungus. So they're just like in this protist group. And it used to be Kingdom Protista. And so, if you look at everything in domain Eukarya, you've got Kingdom Fungi, Kingdom Animalia, Kingdom Plantae, and then the protists. At one time, Kingdom Protista, but now scientists are like, no, there's just too much going on here. They're totally good. There's so much diversity there. There's multiple kingdoms, and it's taxonomists, certain taxonomists are having a good time figuring all that out. So protists, there's a lot of diversity, like I mentioned. There's unicellular, there's colonial, there's multicellular organisms, and then you've got, most of them are aquatic, but we have some that aren't. And if we think about all the protists, some you could look at and you could be like, well, it's kind of animal-like. This one's kind of fungal-like. This one's sort of plant-like. This one's plant-like sometimes, and sometimes animal-like. And so there's all kinds of like craziness happening there. Yeah, sort of a mismatch. So I like to kind of think about it like this, like there's the algae, which are more the plant-like ones. There are protozoans, which are more the animal-like protists. And then there's the water molds and slime molds, which are more fungal-like, but they're not true fungi. There's a lot out there. So a big part of like where you would group them here, what is how they make food, or how they ingest their food, how they take their food in. And some make food by photosynthesis. Some just ingest, like actually have like a special part of their cell that they can act as like a mouth kind of, or kind of like an oral groove. And then there's some that just like absorb the nutrients. And some that just, engulf it. And they have all kinds of different ways of reproduction, asexual and sexual. So When you think about where life started, we all know like life started in water, and a big part of that is because they're protected from drying out, they're protected from UV, and then also a big body of water helps to keep the big fluctuations in temperature down. And then just reproduction. Things, egg and sperm can be sent out, and so water helps with that. This is kind of old. I just like how it's set up. An old way to look at life being split up. Ancestral prokaryote. You've got archaea, bacteria. Ancestral eukaryote that then gave rise to all these different branches. The more fungal protists, the more plant-like protists, the more animal-like protists. And then we have animals branching off there, plants branching off of the green algae, and fungi branching off of the fungal-like protists. We think of like evolution in the tree of life. So this one, we'll start with euglenoids. And the euglenoids are really interesting. Scientists are kind of like, they should be their own, maybe they should be their own kingdom. They're just like so interesting. And there's some that are more plant-like, some that are more animal-like, and some that can like kind of change it up in their lifetime. And their chloroplasts are surrounded by 3 membranes. How about plants? How many membranes are surrounding chloroplastic plants? 2. And so you've got the membrane of the original cell, and then what's left of the engulfing event, when part of the membrane came off of whatever ate that cell. And so when you think about something having more than one membrane, this sort of thing, you have this sort of thing happening. Here is a cell that has a chloroplast in it. And then you've got a green algal cell here, a red algal cell, which would have been like more of the early photosynthesizing eukaryotic cells. And then we've got one of those got engulfed by another cell. So now you've got a chloroplast inside of another cell, or you have a green algal cell that got engulfed, and now it has an extra floral like membrane around it, and then that was taken in, and then it just evolved and became a different kind of chloroplast. And you also see sometimes chloroplasts will change shape in some of these cells. Some are spiral-shaped chloroplasts. Some are flower-shaped chloroplasts. There's all kinds of interesting things happening there. So we see euglenoids have, remember I said 3, so that's that example showing how you have 3. And you might have some things like different diatoms and dinoflagellates, which also have more membrane around their chloroplasts. So we do have euglenoids you're going to look at, euglena. And when you look at a euglena, I want you to look for a couple different things: an eye spot, a flagellum, though they're spinning super fast. They're moving super fast, so they're hard to really identify. See if you can see the nuclei. The other stuff's going to be tricky. You should see some green showing the chloroplast, but otherwise it might be pretty tricky to tell. So the eye spot, it just helps, it has pigments in it that help it know where light is. So if it's like, ooh, I'm getting hungry and there's no food around here to eat, I need to kick into gear and start photosynthesizing. That eye spot can help it be attracted to light so that it knows where it is to move to for photosynthesis. So pretty cool. And then, yeah, so there's some that can photosynthesize for a while, then stop photosynthesizing, and then go back to photosynthesizing, and they're interesting. Dinoflagellates, these make up part of that bottom of the food chain in the ocean. They make up a big portion of phytoplankton. Think about plankton in the ocean, all these little things, tiny things that are swimming around. They make up part of the plant-like part of that. And so they're like the main bottom, but when I say bottom, I don't mean not important. Very important, bottom part of the food chain, providing food for a lot of other things. And they have chlorophyll A and C, and they also have xanthophylls, which provide more like a brownish, kind of golden kind of color to them. They have interesting plates that look like armor that are made of cellulose, like plants. And they have two flagella that when they spin, and sometimes they'll tuck them in these grooves, but it'll cause them to spin. It causes them to spin. They're biflagellates. And some of them can bioluminesce. And sometimes they can lots of nitrogen, certain things in coming out, like if you have wastewater going out into the ocean, or a lot of runoff from agriculture, then these guys can start to really multiply with all that extra nitrogen, and you get a red tide, which can sometimes have neurotoxins build up in it. These guys can also produce neurotoxins. So if you're lucky and you can check it out and you're out in a dark night on the coast, you can sometimes see if there's a lot of them, the waves glowing, or you can rub your hand in the wet sand and see like little sparks happen, and that's these guys getting agitated and woken up. I did order some, but it takes them a little while to kick in, and sometimes they just never work. So at some point, when I'm seeing that they're working, I'll be like, hey guys, let's look at these flagellates, and we'll look at them under the microscope and see if we can get them to glow for us. Source of fossil fuels. There's a lot of plant material, any photosynthesizing material that builds up and settles down low in the earth. We can access that, and that's where we can get our fossil fuels from. And these guys can make up a big part of that. Diatoms is another. Diatoms, along with dinoflagellates, make up that phytoplankton, that really important phytoplankton category of organisms that live in the ocean. These guys do not have flagella like the dinoflagellates do, and instead of having these cellulose sort of like plates on them, they're actually made of silica, glass. And their little silica glass, their glass houses can be really intricate and beautiful, all kinds of different shapes and textures, designs. They also have chlorophylls A and C, and they have a more brownish pigment in them called fucoxanthin. They can be found in fresh and salt water, and you get a buildup of these guys, and they can make what's called diatomaceous earth, which is like a white, chalky material. Let's see if we have a little bit out here. Oh, we don't have those out yet. Where did our diatoms go? Our stuff is just getting moved around a bit. Here we go. So when you look at diatoms, we do have some. We're just going to scrape a little bit of this diatomaceous earth off onto a microscope slide, add a couple drops of water, good to go. I mean, you need a tiny bit. If you get too much, it's just like, so dense you can't really see the shapes. So try to just get a little bit of this stuff. So let me test that around. It's okay if you get a little bit on you. You just don't want to breathe a lot of the dust in because they're little tiny sharp pieces of glass. You don't want to get them. Yeah, you don't want to get them in your lungs. So they're used for abrasive filtration. This stuff's used in pool filters. It really works great for filtering out things. So it's a sedimentary rock. And I'm not going to have you guys memorize. You do have to memorize life cycles of plants. This is botany, right? Makes sense. I'm not going to make you memorize this life cycle, but I just think this is so cool. So we'll share this, and then you're gonna go to break. So this looks like a petri dish, right? Doesn't it look like a petri dish? That's exactly kind of how the general shape is. But sometimes you can have some diatoms that are like long and skinny, or sometimes they're like a true petri dish shape. But they have these two sides to them, where one's bigger than the other, and one fits over the other. And they can break those sides apart if they want to. And they can do that for asexual reproduction. So you're a diatom, you're like, I'm gonna reproduce. And it can break itself apart, and then it makes a new, each side makes a new half. So if here, starting with this one, if that breaks apart, then that bigger side is gonna always be big. As it breaks apart, it makes a new, smaller half that fits in it. If you're the smaller side, and you make another half, and you just keep staying that half, you'll stay that size. But if you split, and then make another half, and split, and then you end up being this little half, you get too small to the point where it's like, oh, I'm too small. I'm gonna undergo meiosis, and then sexual reproduction occurs at that point. But all along, it's asexual reproduction. Isn't that crazy? So I think it's pretty interesting. So then you've got gametes being produced, zygote, and then starting out again as the big form. And so it progressively gets smaller and smaller and smaller, depending on which half is splitting off and which one's splitting off. Yeah.
Is it actually 3 generations before it starts doing meiosis, or is it variable? Does it vary?
Oh, that's an excellent question. I'm guessing it varies depending on the size, but I don't know. That's something we'd have to look up. Yeah, let's look that up. Let's check that out. All right, let's go ahead and stop there, and we have just a little bit more to talk about, and we'll jump into it. So when you come back, you can grab your microscopes just to get that up.